I am Bernard Herschel. Before my retirement, I was the chief of HIV/AIDS at the Geneva University Hospitals. You may call me an AIDS dinosaur who has followed the HIV epidemic from the very first cases in the early 1980s through the triumph of antiretroviral therapies, a triumph unequaled in the annals of medicine. This triumph has its flip side. Younger colleagues are no longer familiar with the opportunistic diseases and the myriad manifestations, hence the idea of these videos. After a short introduction, they will show a series of images illustrating one or several exemplary cases from my files. Leishmania are protozoans. It is single-cell microscopic animals. They exist in two forms as mastigotes, that is, motile forms with a flagella, and amastigotes, defined as mastigotes having lost their flagella. Promastigotes are the precursors of the free-living mastigotes. See the details at left, but note that the drawing is not to scale. An amastigote measures approximately 3 × 2 microns, whereas the promastigote without the flagella measures approximately 10 × 2 microns. Life cycle of Leishmania, where we distinguish sandfly stages, red arrows, and human stages, blue arrows. When the sandfly takes a blood meal, it injects promastigote stages into the skin. The promastigotes are phagocytized by macrophages and transform into amastigotes. The amastigotes multiply in cells of various tissues. When then the sandfly takes a blood meal, it ingests these macrophages infected with amastigotes. The amastigotes transform into promastigote stages in the midgut of the sandfly, and these promastigotes migrate to the proboscis, whence they are injected into the skin of various hosts. The minuscule sandfly is depicted on the left. On top, a section through the midgut of the sandfly with abundant Leishmania promastigotes adherent to the brush border epithelium. We can distinguish 4 forms of Leishmaniasis. Cutaneous Leishmaniasis, traditionally associated with the Middle East, is characterized by skin ulcers. Mucocutaneous Leishmaniasis causes destructive ulcers and is seen in the Middle East, India, and South America. Generalized Leishmaniasis, or kala-azar, causes pancytopenia, hepatosplenomegaly, weight loss, and fever. Finally, post-kala-azar cutaneous Leishmaniasis is characterized by skin papules. The last 2 forms are those which are associated with HIV infection. Cutaneous Leishmaniasis. This is called in French the bouton d'Orion and in English the Jericho button. On the left, 2 images of US soldiers in Iraq in 2003, and to the right, a British soldier in Palestine, 1917. Global distribution of the different types of Leishmaniasis: visceral in green, cutaneous in red, visceral plus cutaneous purple. It should be noted that all forms can coexist in the same locations. In Europe, Leishmaniasis HIV co-infection occurs almost exclusively in those who have acquired HIV through IV drug use. Rarely, Leishmaniasis has also been transmitted by transplanted organs, pointing towards the existence of latent carriers. The patient was born in 1955. He was an IV drug user in the late 1970s and started methadone maintenance in 1983. He was diagnosed with HIV in January of 1999 with a CD4 count of 92 and 103,000 HIV RNA copies per ml, treated with Norvir, Crixivan, and D4T, and also with cotrimoxazole for primary prophylaxis of PCP. He had an excellent control of his viremia, but the CD4 counts remained stubbornly below 200. Now let's turn to the Leishmaniasis side of this patient's history. In June 1998, that means before the diagnosis of HIV, he presented with pancytopenia, hepatosplenomegaly, and a diagnosis of disseminated visceral Leishmaniasis was made with positive biopsies of the duodenum and bone marrow. He was treated with antimony derivatives for 28 days. Lengthening of the QT interval was noted. In November 1999, he had a relapse diagnosed by duodenal and esophageal biopsies, and this time he was treated with amphotericin B for 28 days, complicated by hypokalemia. In July 2000, he had a second relapse diagnosed by bone marrow biopsy. He was treated with liposomal amphotericin followed by oral miltefosine. But in December 2000, same story, and this time after treatment with high doses of liposomal amphotericin B for a total of 2 grams in 5 weeks, maintenance treatment with amphotericin was tried. We are now in September 2001, and again the patient complains of loss of appetite and weight, of fatigue, and of a skin rash. The medical chart records ankle edema, a palpable spleen, and papules and pustules on the face extending to the neck. Laboratory results show pancytopenia and polyclonal hypergammaglobulinemia. HIV replication is well controlled, but the CD4 count is still low 2 years after starting HART. Biopsies of skin and esophagus, as well as gene amplification, confirm the relapse of Leishmaniasis. An overview of the patient's complicated history with multiple relapses and treatment attempts. LAB equals liposomal amphotericin B, SB, pentavalent antimony, PKCl post kala-azar cutaneous Leishmaniasis. Note that the CD4 counts remained low for 4 years until 2003. The character of the Leishmaniasis gradually evolved from disseminated visceral Leishmaniasis to post-kala-azar cutaneous Leishmaniasis. Relapses ceased only after 2003, when finally the CD4 count rose to near normal levels. Post-kala-azar cutaneous Leishmaniasis, or PKCL, is a chronic skin rash observed following clinical response to treatment for visceral Leishmaniasis. There are erythematous or hypopigmented macules that progress to plaques or nodules. The lesions resemble and must be differentiated from those of leprosy, but are not associated with sensory neural changes. The papular non-pruritic rash of post-kala-azar cutaneous Leishmaniasis. Partial clearing after a course of therapy with pentavalent antimony and miltefosine. Histology showing inflammatory infiltrates in the skin and subcutaneous tissue. At higher magnification, the mononuclear cells appear granular, and a touch preparation of a skin biopsy colored with the Yemsa stain reveals Leishmania in a monocyte. Leishmania may be dangerous, but they are also beautiful. Here from the bone marrow at higher magnification. Humans are not the only animal infected by Leishmania. An immunofluorescent stain used to diagnose Leishmaniasis in a dog. And in tissue culture, DNA in blue, Leishmania in green. These two images show Leishmania pro-mastigotes invading human macrophages. Chagas disease and Leishmaniasis are both vector-borne, HIV-associated, and caused by related prokaryotes, Trypanosoma cruzi for Chagas, Leishmania for Leishmaniasis. The distribution is a little different. Chagas disease is only seen in South Central America. The vector is different, kissing bugs for Chagas disease, the sandfly for Leishmaniasis. In Chagas disease, it's the heart and the nervous system that are involved. In Leishmaniasis, the skin, the liver, and the spleen. The typical presentation of Chagas disease in persons living with HIV is the chagoma, a cerebral mass. This patient was a 29-year-old woman who was 26 weeks pregnant. She had Trypanosoma cruzi parasitemia and a cerebral mass. The PCR for Trypanosoma cruzi in the cerebrospinal fluid was also positive. This HIV-positive patient presented with hemiparesis. The CT showed a cerebral lesion with surrounding edema and marked mass effect. The differential diagnosis would have to include glioblastoma, toxoplasmosis, and cerebral lymphoma. Biopsy confirmed the suspicion of chagoma. T. cruzi A. mastigotes in the brain biopsies of HIV-positive patients. In the image at right, the insert shows a kinetoplast. Talaromyces marneffei was called Penicillium marneffei until 2015. Talaromycosis is the new name for penicilliosis. With HIV infection, symptoms are secondary to hematogenous dissemination. It is fever and loss of weight, enlargement of liver and spleen, umbilicated skin lesions, respiratory symptoms, and blood cultures are invariably positive. In the rare patient who have talaromycosis without HIV infection, there is more frequently bone and joint involvement. In Thailand, Taloromyces marneffei is the third cause of opportunistic disease in AIDS. Cases have also been reported from southern China, Hong Kong, Cambodia, Vietnam, the Philippines, Malaysia, and Singapore. The bamboo rat is the natural host of P. marneffei, but it is unknown whether it plays any role in the transmission to humans. This graph is from Chiang Mai, Thailand. It shows the concomitant rise and fall of HIV infection on one hand and P. marneffei infection on the other. T. marneffei is dimorphic, showing pigmented colonies at room temperature and white, chalky colonies at body temperature. Dimorphism is also apparent microscopically, with mycelial forms at 25 degrees, top left, and yeast-like forms seen in a silver stain of a tissue section, top right. Note the transverse fission, which helps to distinguish T. marneffei from other dimorphic fungi, such as histoplasma. T. marneffei can be detected in the bone marrow, top left, tissue sections, top right, and skin and lymph node imprints. Specific fluorescent antibody staining over skin scraping is shown at left. 3 microorganisms which can be detected in skin scrapings: Histoplasma capsulatum, Leishmania infantum, and Talaromyces marneffei. Papules and umbilicated lesions caused by T. marneffei, with close-up at right. Lesions due to T. marneffei in an HIV-infected child from Chiang Mai. These lesions look very similar to those caused by Molluscum contagiosum, here in an AIDS patient in Geneva. 2 further examples of molluscum-like skin lesions due to P. marneffei in Chiang Mai. Hemorrhagic and ulcerated skin lesions due to T. marneffei. Umbilicated skin lesions in AIDS patients have diverse causes, as shown here: histoplasmosis, molluscum, cryptococcosis, and T. marneffei. T. marneffei skin lesions before and after treatment with itraconazole. Thank you for watching, and thanks in advance for your feedback. This video is part of a series on opportunistic diseases in HIV/AIDS. If you are interested, please subscribe or click on the latest upload top left.